everyone. I think we're going to get started. Welcome, welcome. It's great to see you all here tonight as we open the exhibition Ice Dinosaurs and say goodbye to the exhibition Life Onto Land, which I hope all of you have had a chance to see. If you haven't, it closes on Sunday, so be sure to get up there tonight and make sure to tell all your friends. We'd like to extend an especially warm welcome to our members for your passionate belief and support, which makes all that we do here at the Academy possible. I'm Marina McDougall, the Vice President for Experience and Engagement, and I've been anticipating this evening's program, A Transforming Earth, the Legacy of the Devonian Period, with great excitement. 2024 has been Evolution Year at the Academy, a year in which we've focused our programming on evolution, the remarkable, ever-unfolding, existentially fascinating story of all living things, including us. The Academy's core work in systematic biology and evolution over the institution's 200-year-plus history involves the careful work of piecing together the story of life on Earth. The more than 18 million specimens in our collections are vital to the global project of reconstructing evolutionary patterns that inform today's critical conversations and efforts to address climate change and biodiversity loss. While the periods of the Jurassic and the Cretaceous are well known in popular culture, the Devonian, not so much. But we aim to reverse that. Life Onto Land, the Devonian, a key project for the Academy's Evolution Year, shines a light on a fascinating and arguably more consequential period of geologic time, spanning 60.3 million years ago, 419 to 359 million years ago, also known as the Age of Fishes. During the Devonian, the first forests emerged and the first animals began to crawl onto sandy shores. It was a time when our modern terrestrial ecosystems first began to take shape. The exhibition centers around and celebrates some 30 years of research by the Academy's amazing vertebrate paleontology team under the leadership of Dr. Ted Daeschler. Ted, you want to wave? Great. Working in close collaboration with research associate Dr. Jason Downs, fossil preparator Fred Mullison, and collection manager Ned Gilmore, along with Drexel students and colleagues. Some maybe are here tonight, and colleagues from around the world. The exhibition also includes materials from the Academy's invertebrate paleontology collections, botany, malacology, and ichthyology. We're also honored tonight to be joined by one of our international partners, Scott Ruffalo, paleobiology curator at the Canadian Museum of Nature in Ottawa. Scott graciously facilitated the loan of the world-renowned holotype Tiktaalik rosé with the agreement of the Nunavut peoples. For tonight's conversation, we're delighted to be joined by scientists Dr. Diane Boyer and Dr. Jonathan Wilson, who with their expertise in ancient marine communities and the evolution of terrestrial plants will help us to gain a more holistic picture of the Devonian and its many, many meanings to the world today. Before we start, I'd like to give a special thanks to our public program manager, Molly Gross, who organized tonight's program. We have John up in the booth, and our facilitators, Desta and Denise, will help to ensure that your questions are answered during our conversation. Jason Downs, our longtime friend to the Academy, a vital contributor to the vertebrate paleontology department, as well as the development of Life Onto Land, will introduce our guests and moderate the conversation. So please welcome him, and again, thank you all for being here tonight.
Thank you, Marina, for the introduction. Thanks to all of you for coming out. This is an exciting evening for me I'm a research scientist here at the Academy of Natural Sciences, but I was also on the development team for the Life Onto Land exhibit. And that was the realization of a dream that Ted and I had for many decades, to put the research that we do upstairs out on display for the public. And it saddens me to know that the exhibit is coming to an end, as Marina mentioned, but tonight I'm excited because this really is an opportunity to bring the Life Onto Land exhibit to life in a new way by the presentations that you'll hear, and also the discussion that we'll have afterward on stage, and then also expanding the conversation out to include all of you. So I'm excited to get started. I won't waste any more time. I'll begin by introducing our first speaker. Our first speaker is Dr. Diana Boyer from Winthrop College. She's a professor of geology there, and I'll ask her to come to stage.
Thank you. I'm also very excited to be here tonight. And so what I want to do is start with a little bit of an introduction to myself, and maybe a little bit of an introduction to the Devonian, and hopefully you will all leave here as raving fans if you're not already. So I am a paleoecologist, and so what that means is that I like to try and reconstruct ancient communities. And so taking fossils to understand what organisms lived and how they interacted with their environment, those are the things that kind of get me excited. But I want to take a minute and remind us all that during the Devonian, some 370 million years ago is where this reconstruction was from, the entire planet looked different. And so even just the continental configuration was different. Climate was much warmer, sea levels were higher, and a lot of these light blue areas of the flooded continent allowed for fantastic reef ecosystems. And so this continent here is actually North America that's shifted to the side. And here in Philadelphia, we probably would have been somewhere close to there. So certainly underwater for most of the Devonian. Another thing about me is I really quite enjoy the field aspect of it. So where do you get the information to try and build and understand what conditions were like during the Devonian? Well, you have to start with the rocks and the fossils. And we can learn so much from not just looking at the shape of the organisms, but even the chemistry of the rocks to try and understand what the oceans were like long ago. And I would also emphasize, I work with collaborators from all around the world, but you don't have to go all around the world to find some fabulous Devonian rocks. So here I'm out in Western Utah with some of my students and collaborators, but even in places like upstate New York and Northern Ohio and here in Pennsylvania, you can find some fabulous exposures. So you don't always have to go to exotic locations to investigate these. Now, I am a paleontologist, and usually when people find out, oh, you're a paleontologist, they immediately think dinosaurs, right? And then the disappointment comes in when I say, I actually don't work on dinosaurs. And so I'm a paleoecologist reconstructing environments, and I really like the stressful situations. Low oxygen, dramatic climate changes, and actually I'm working on the late Devonian mass extinctions, so that's what gets me really excited. And under really stressful conditions, I know this is a little bit twisted, right? Under these really stressful conditions, organisms often don't have the extra energy to expend to build a skeleton or make a shell. And so these stressed out communities often are made up of soft-bodied, worm-like critters. And the evidence they leave behind are called trace fossils. So I'm a paleontologist, and I work on worm burrows. But they can actually tell us about the types of organisms, the size, the diversity, the abundance, and that coupled with the chemistry can help us understand the causes and consequences of major transitions in life during the Devonian. I also have to point out some of my favorite friends. Brachiopods here, they're shelled critters, and we've all collected shells on the beach, right? And most of the time they're mollusks, clams, oysters, snails. But if you went beachcombing in the Devonian, you would find completely different organisms. There's still shelled creatures, invertebrates living in the shallows, but they're going to be all different types of brachiopods. And in fact, the differences in the biota don't end with just the invertebrates. Devonian had extensive reefs. So the ecosystem was incredibly widespread, developed, went to much higher latitudes, But the organisms making the reefs were not the corals we know today. They were these layered, sponge-like creatures with a fantastic name, stromatoporoids. And so these are some of my favorite fossils that were dominating during the Devonian, and hopefully we'll get a little chance to talk about. So I will.
Great. Thank you, Diana. Our next speaker is going to be Dr. Ted Daeschler, who is Curator Emeritus at Drexel University in the Academy of Natural Sciences.
Thank you all for being here. It's really fun for me to be back up on this stage. I retired about a year ago from the Academy, but the Devonian is in my blood, the Academy is in my blood, and I've been upstairs on and off, even in my retirement. I was here for 36 years and a professor at Drexel for 11 years at the same time. And so I've been really lucky to have these opportunities right in this building. So this is me in my natural habitat, or should I say preferred habitat. This is about 2 months ago during exploration for Devonian age rock and fossils on Ellesmere Island in the Canadian Arctic. And my friend Scott Ruffalo from Canadian Museum of Nature was there with us. It was an exciting trip for many reasons. We found some terrific fossils. It was an adventure. We had some terrible weather. We had some interesting plane flights, and helicopters couldn't come and go, but that's all part of it. And the excitement of discovery makes all of the hardships and the ability to be in places like this make it all worthwhile. So paleontology is definitely a process, and there's many stages to that process. And there's many different kinds of activities and really different kinds of scientists. Field work is one of my most favorite parts of paleontology. That's getting out to places like this on Ellesmere Island, not only for discovering new fossils, but also geological work to establish the environmental context of this ancient life. And therefore the sort of the evolutionary pressures which may have been helping to create and other group, some forms diversifying, some forms going extinct. So fossil collecting represents new data many times, and it's an exciting part. And then collection building, whether it's bringing fossils here to the Academy of Natural Sciences, fossils from Canada will go to the Canadian Museum of Nature, where they hold fossils that are part of or from the Nunavut territory. And collections in places like the Academy and elsewhere ensure that these fossils are prepared and preserved and studied by generations of students and professionals, and also put on public exhibit, particularly for education. But again, our work doesn't have to be far away. Our work on Devonian vertebrate fossils started and continues much closer to home. This is one of the most important late Devonian non-marine sites, not only in Pennsylvania or North America, but maybe the world. And it's on a highway roadside, just a few hours drive from here. We call this place Red Hill. It has produced a wide variety of vertebrate fossils, as well as many plants and invertebrates, all within the ancient stream deposits of the Catskill Formation. And we've described numerous new species from these deposits, including some of the earliest known limbed vertebrates, so the animals we colloquially call tetrapods. And they seem to have evolved in response to the development of these sorts of freshwater stream environments. So here's a reconstruction of some of the organisms from the Red Hill site and the sorts of geological settings which are represented by those sedimentary rocks. And really, more generally, across the globe in these really in the Devonian burgeoning freshwater ecosystems. And I think you can probably sit back and relate to this sort of place. We recognize streams and swamps and forests and the type of life that are supported in those ecosystems. But importantly, this environment and those ecosystems it supports did not exist on Earth until the Devonian period. So that's more than 90% of Earth history. This kind of place and the life it supports did not exist. You could not have found that. So if you go back in time, you want to make sure you go from the Devonian to the present, otherwise you're going to have a hard time finding a place like this. It's very clear then that the evolution of plants, animals, and environments during the Devonian really became the crucible for many evolutionary innovations. And the innovation that I'm particularly interested in is the development of terrestriality in vertebrates, the movement onto land. Look forward to talking more about it.
Great, thank you, Ted. And our third speaker is Dr. Jonathan Wilson, who's a professor of geology at Haverford College.
Good evening, everybody. I am a paleobotanist, and the way I like to explain my research to people who are sitting next to me on an airplane, for example, is I study fossil plants, but what I like to do is I like to take fossil plants and I like to run them on treadmills, metaphorically, and figure out how fast they could grow, what environments they could live in. And we're really fortunate as paleobotanists because plants all do the same thing. Plants all trade water for carbon to conduct photosynthesis, and they're a real pipeline of energy from the environment into the biosphere. And yet, there's a world before and after land plants, and this transition happens during the Devonian. The Earth changes from a world where we have small, scrappy terrestrial organisms to a world with forests and wetlands and trees so large that if you could travel back in time with a chainsaw, you could cut it down and build a tree out of them. For those of you that are familiar with the history of animal evolution, the Devonian is like the Cambrian explosion, but for plants. We have a rapid evolution of all of the major groups of land plants, seed plants, ferns, lycophytes, and bryophytes. And we see huge advances in morphological complexity over these 60 million years. We go from plants that, if I show a fossil to somebody they're like, that's actually a pin, that's not really a plant, to a leaf that's recognizable as a leaf that you might see on a tree in your backyard. And these changes in turn have dramatic effects on the Earth's system. They connect and alter and drive environmental change around the globe, from providing new habitats to changing atmospheric chemistry to even changing the chemistry of the ocean. And this is really a profound state change for the planet. There's a before land plants, and then there's after land plants. What I've got here for you is a sort of reconstruction that merges some of our earliest land plants. Oops, sorry, let me go back. Go back just a little bit, there we go. That merges our picture of early land plants with some later land plants from the Middle Devonian here. And at left, we have some scrappy non-vascular plants. These are things like mosses. In the middle are some early vascular plants that are here that have these sort of football-shaped sporangia up at the top. And then at right, these are the plants that are the ancestors of our ferns and our seed plants. But all of these are kind of variations on a theme. They're all green photosynthetic sticks. But these subtle differences in their morphology go on to have profound meaning for ecosystems around the world. By the end of the Devonian, we have tree trunks with branches and leaves that look more exciting and modern. And we're really fortunate in the Devonian period to have a couple of extraordinary deposits that give us these tantalizing glimpses into plants colonizing the land. For example, Red Hill is a very famous one, a beautiful deposit. There's another one near Aberdeen in Scotland called the Rhynie Chert, which preserves an early Devonian Yellowstone ecosystem. And it preserves everything from the ecosystem together: plants, insects, fungi, even insects feeding on plants in these early ecosystems. And then we get these extraordinary finds like this one here. This is a late Devonian tree trunk of Archaeopteris. It's washed up on a beach in what's now Morocco. And it's just like a giant chunk of driftwood, but from driftwood that landed on a beach 370 million years ago. And note this rock hammer for scale, it's a massive tree, and this is Archaeopteryx, the first modern tree. And by assembling these fossils, these fossils give us glimpses of what the whole world looks like. And what's inside the plants is as interesting as what their shape and size tell us. We have a really rich record of plant anatomy, and these are a series of photos of internal anatomy of plant fossils, roughly from oldest to youngest, all from the Paleozoic era here, from simple to more complex. And what I want to point out to you is the first 5 images here are from the Devonian. We start out with very simple internal anatomy, and then we start getting more complex shapes. And all plants function the same way, therefore we can actually quantify and measure how quickly these plants could grow, what kinds of drought stress they could survive, and how resilient they are. And the message that we have today is that these plants are not primitive at all. They're doing some extraordinary things, and some of the features that we associate with modern plants, large size, fast growth rate, extreme drought resistance, actually first appear in the Devonian period in these early ecosystems. This is a particularly exciting time in plant evolution for me personally, but looking at this study here. And we have these tantalizing clues to the history of plant evolution and the history of the planet, but there are also some very interesting mysteries to be solved. And I think I'll leave that for the discussion.
Thank you, Jonathan, and thanks to all 3 of you. I think what each of you touched upon is the Devonian period as a time of transition, and I think Marina described it as an especially consequential period of geological time. So I'd like to hear each of you elaborate on the Devonian period as this time of transition, using the different kinds of data that are of interest to you. And I think about framing this conversation in a way that treats biology on different scales, these different scales that we can think about it, from the molecular chemical scale up through cells, through anatomical interactors, up even to sort of which species are dominating these spaces at the start of the Devonian versus the end of the Devonian. So if we think sort of small scales first, would anyone like to sort of talk about what we can learn from the smallest scales of biology in terms of kind of understanding the transitions of the Devonian period?
Yeah, thanks, Jason. I can start. What I work on for my research is actually, I apply fluid dynamics models to the vascular systems of extinct plants. So these are subcellular structures. These are usually about 50 microns in diameter, so they're about half as wide as a human hair. These structures are very small in land plants. And one of the things we know from observing these from our earliest land plants to the latest land plants in the Devonian is that plants very early on evolve this ability to transport as much water to their photosynthetic surface as possible. So it's getting as much energy to the site of evaporation to absorb as much carbon and grow as quickly as possible. And in different lineages, we see different plants sort of reaching this strategy in the same way. And yet, by about 10 million years into the Devonian, we start to notice that plants start to develop adaptations to resist drought. They can no longer survive this kind of live fast, die young, profligate lifestyle where it's just get as much water and survive if you can. And we start to see these adaptations appear that allow them to be more resilient in the environment. And those turn out to be features that are carried through right to the present day. And so on the one hand, if you just look at these early land plants and you look at them, you're like, wow, what a risky choice you've made. But on the other hand, they're experimenting with these physiological combinations that shed some light on why certain things like pine trees or certain kinds of lycophytes are incredibly resilient today, despite having this long evolutionary history.
So I might go to the other side of the scale, and just because I do marine work, the Devonian's dominated by these reef ecosystems in the shallow marine settings. And so one of the things that serves as a real transition and a really interesting question that we can look at and evaluate from all different scales is, how did these reef ecosystems get so big and so expansive, and who were the players that made up these ecosystems? And then what happened to cause their complete demise? There was a massive, one of the biggest 5 mass extinctions in the history of our planet happened towards the end of the Devonian. And what caused this? And in investigating who made it and who didn't, who were these live fast, die young, and who were the ones that had developed systems and behaviors to survive these events is really interesting. And we definitely investigate these on a range of scales, from the anatomical level using geochemistry to the hand specimen up to mapping large reef systems.
I think my comments when Jason asked the question, what I thought about was sort of the tree of life, okay, all invertebrate life, which is what I've been focused on. You know, we can see the first vertebrates, very simple forms back into the Cambrian, and then the Ordovician and Silurian. These are the periods that precede the Devonian. And they're dominated by things that, you know, if we saw them today, you'd think that is just a weird animal of some sort. These, we call them anathans, jawless fish, you know, just a lot of other things, these armored fish that we colloquially call placoderms, sort of early experiments in aquatic finned animals. But what makes the Devonian so consequential, I think, in the story is that, so those early forms are doing their thing, you know, they're there, they're abundant, they're making a living, they're contributing to ecosystems, marine ecosystems primarily. But what you see in the Devonian is sort of a switch to sort of out with the old and in with the new. And the groups, for example, chondrichthyans, the sharks, and the osteichthyans, the bony fish, particularly ray-finned fish, which is most of what we think about today as fishes. Those are both getting a start, and sort of course, starting slowly, but by the end of the 50 or 60 million years of the Devonian, becoming more common. Then, of course, the group that we tend to focus on, because we're part of it, are also bony fish, but the lobe-finned lineage of bony fish. And they diversify in all different directions, and one part of that group starts to specialize for shallow freshwater environments, that crucible I mentioned earlier. So I just see the Devonian in a big picture way as being out with the old and in with the new.
Yeah, and I think this also speaks to the legacy of the Devonian period, which is part of the title tonight, which is, you know, kind of thinking about the Devonian period as, our exhibit bills it as such, the dawn of the modern terrestrial ecosystem. But I don't think it's just isolated to the terrestrial space, but there are changes that are happening in the Devonian period that turn the biosphere into one that we continue to recognize today, that some of these changes make this biological space the same one that we can recognize when we go observe it today. So maybe this is sort of pushing us in the direction of thinking more about the end Devonian extinction, but you know, does anyone want to sort of talk about how the Devonian changed the biosphere in a way where it, you know, it kind of turned it into the one that we recognize today in a lot of ways? And you're speaking to this a bit, Ted, where you mentioned that, you know, jawless vertebrates, for example, are incredibly diverse in the Devonian period, but most of those lineages don't make it out of the Devonian period. So we have hagfish and lamprey today, but really vertebrates have jaws, more or less. So yeah, I think it's interesting to think about the Devonian that way as well.
I think I'll make a very short statement, which is I give all credit to plants. Yeah, if plants hadn't sort of started that parade into the terrestrial ecosystem, at least, nothing else would have happened on land. So,
on behalf of plants, if I can accept that accolade, I'll gladly accept it. And actually, I think you know, plants are this interesting interface in the Devonian where they're both providing habitat, but I think there's been increasing evidence that this terrestrialization of vegetation is starting to change kind of fundamental Earth science, Earth system processes. It's starting to change runoff into the ocean. They're starting to change the energy balance because solar energy isn't hitting bare rock. It's starting to hit forests, and that's going to change precipitation patterns. And so I think plants may be not only providing habitat for these organisms to terrestrialize, but they may actually be causing some pretty important changes in the ocean as well. And maybe I'll pass this to Diana to talk about.
So first I want to acknowledge, I think you're right, the Devonian does not get enough love. I think it is this kind of like hidden, really important time. And I think you even forgot to mention the development of soils. And so the plants really do, they change the entire terrestrial, atmospheric, and oceanic chemistry. And so chemistry can be boring, but if we just think about carbon dioxide. So carbon dioxide is utilized in photosynthesis, is going to change temperature on this planet, and the runoff and the carbon sequestration, and all of this is going to change really all aspects of life. And so thanks a lot, plants.
But continuing to think about reefs, reefs are having a hard time of it right now with modern climate change. And so, you know, what do you see if we look at reefs at the start of the Devonian, through the Devonian, and then across the extinctions that happen at the end of the Devonian?
Yeah, and so I think, I mean, we have to remember, we've run these experiments before, and we have a record of it. We can look at the rocks and see, well, what happens if we cause major climatic perturbations? Like, what happens if we raise the temperature dramatically or decrease it? And it has a major impact on life on this planet. And so we can look at the late Devonian, we can look at this run up and see what characteristics, what critters, which environments were most successful. And then we can see when you change things, and there's a huge climatic shift at the end of the Devonian, what happens? Well, we can go look in the rock record and see who the winners and the losers were and how bad it can get and what the recovery can look like as well.
And Ted, shifting things to you, I think one of the things that has always interested me in the work that we've done on lobe-finned vertebrates is that the transition, between kind of fin and limb, and, it's referred to as the fin to limb transition, the terrestrialization of vertebrates, it really is a transition that is felt or it's observed throughout the entire body. So could you talk a little bit about terrestrialization beyond just the obvious, which is fins into limbs?
Great, glad to.
Oh, I do. May just be low batteries.
Yeah, it's a little counterintuitive when we think of, we sort of shorthand say fin to limb transition, the origin of terrestrial life. Let me start by saying there were limbs before there was terrestrial life. That's particularly counterintuitive. So the very earliest of the limbed vertebrates, those early tetrapods, especially the ones in the late Devonian period, they were still tied to aquatic settings. But as often happens in evolution, the structures that we're developing for one function, and we hypothesize that it's shallow water navigation, getting into swamps because there's new habitat, new prey items, getting away from the predators that are in the deeper water, whatever it might be, those appendages were quite fin-like. They were becoming something that we would call a limb, but they weren't for, running around on the dry land yet. That would come, right? Although we have plenty of limbed animals that are aquatic today. Think of amphibians, salamanders, and those sorts of things. So anyway, but then indeed, there's a lot more than locomotion, a lot more than moving. There's respiration, there's prey capture, all these different systems that need to change as well if you really are going to fit into a new ecological opportunity, that opportunity being more and more terrestrial living. Moving around is a part of it. And of course, limbs are important, but so's the backbone, So is the neck and the musculature associated with all these other parts of the body. So when we do find an early limbed animal, sometimes we don't even find the limbs, but we find really interesting stuff throughout the rest of the body, which is helping to put together that picture of, well, what were these animals specializing in doing? How were they fitting into ecosystems that we're trying to reconstruct? And so there's just so much to learn there.
And remarkable then I know it's been the case that the discovery of a single lower jaw or a single skull bone is often enough to infer that limbs were present in this organism, because it isn't just sort of, a body losing the fin to gain a limb. It is something that is experienced throughout the body.
Well said.
Yeah.
Yep.
And shifting over to you then, Jonathan, if you think about terrestrial plants at the start of the Devonian to the end of the Devonian, it like thinking about the Devonian as a time of transition, maybe plants illustrate it the best, in the sense that, as you described early in the Devonian, we have these low-lying horizontal stem things that are barely recognizable as plants, more branch-like than the plants we think of today. But then there are trees and forests and evidence of forest fires by the end of the Devonian. So I know, we have this artwork here to represent a Devonian landscape, but, it's like if you were to depict an early Devonian landscape, it would look completely different from a late Devonian landscape. So you've talked about vascular tissue, but even these low-lying plants at the early of the Devonian have a way of moving water around. So could you talk about sort of the Devonian, the transition in terms of just how water is moved around and how these new kinds of tissues enable greater distances of travel? And
yeah, absolutely. This is, plants are faced with this deep fundamental constraint, which is that plants cannot absorb carbon dioxide from the atmosphere for photosynthesis without losing water. And so water is a requirement to conduct photosynthesis, to conduct this metabolism. And so plants, if you look at the history of plant development, that was a fundamental constraint on early plant growth. The physics of water movement in cells and capillarity, for any of you that may have had to do those kinds of experiments in middle school or high school, limits you to a very, very small size until plants develop the ability to harness the drying power of the atmosphere to pull water through them. So the plants develop a vascular system that's just a plumbing system. It's just a network of tubes. And as plants open their stomata to expose their wet interior to the dryness of the atmosphere moves water through them. And very quickly, plants evolve very complex shapes of internal structure to kind of maximize and move water to the sites of photosynthesis as quickly as possible. And so as we look at plants start to get larger, their internal structures start to get more complex. And for about a century, this was a big mystery in plant evolution, was why does their internal structure get so complex so early? And it turns out that this is probably adaptations that led to increased drought resistance. So plants started to become more complex to be able to do more photosynthesis. And this happens quite early in the Devonian. And by the middle Devonian, we start to see plants with shapes like this that are very alien to us today. If you go to the tropics today, we're dominated by angiosperms, by flowering plants that evolved in the last 130 million years. But by the middle and late Devonian, you start to really see some unusual trees. They look like something from Dr. Seuss or Edward Gorey or something like that. They do not have leaves, but they have these complex internal vascular systems. And so they have these adaptations to basically move water to these sites of photosynthesis. And then by the end of the Devonian, plants get so large that something in the environment triggers multiple lineages of plants to evolve flat leaves at the same time. And so plants have evolved this plumbing system, and then something seems to happen, and in different plant lineages at the same time in the Devonian, all their leaves get flat and they start to laminate. It looks much more like a modern leaf. So it goes from like a claw world in the middle Devonian to a leaf world in the late Devonian. And all this is based on the fact that plants are just doing basically one big thing. They're moving water to a site of photosynthesis to gather carbon. And this complexity, this kind of escalation interaction that we see is really vivid in the Devonian. As soon as plants crack the ability to develop vascular tissue, all of a sudden the size constraints are taken away, and you start to see plants that quickly go from being just a few inches tall to a foot or two, to a meter or two, to a sizable trunk. And it's really something spectacular to see.
It's a remarkable time. Each of you touched on field work in your presentations, and so we're collecting different kinds of data in different places, and each of us is using a different type of sample. Maybe different preservational conditions are required for each of the different things that we're interested in. So could each of you sort of talk more specifically about the nature of your field work and what collecting is like, and where exactly you're doing it? You touched upon it already, but I'd like to hear more.
So I have spent a lot of time working on, again, these stressful conditions, looking at, often it's preserved in black shales. And so I have been to, creeks in New York, and actually the Cleveland Metro Park system is fabulous, and if you get permissions, they expose fantastic Devonian aged rocks. But, from urban areas to out in the middle of the Great Basin. And what I typically do is I collect hand samples. I use a lot of duct tape because it's friable, it falls apart. And so I duct tape them, and then when you cut perpendicular to bedding, you can see all the little worm burrows or trace fossils. I also take samples to powder for a bunch of geochemical techniques. And so using mass spectrometry and all sorts of sophisticated equipment, we can actually look at the chemistry of the rocks, and it can tell us things like how much oxygen was in the ocean, what the productivity in the ocean was like. Were there a lot of things photosynthesizing in the surface, or were there not? And this helps us tell the story of extreme stress, what the critters were doing, and I will point out that some of this geochemistry, where we look for the productivity in the ocean, requires also a lot of tinfoil because they're looking at the organic carbon, and so you can't use plastic because that is made from petroleum derivatives. So it can be quite a tinfoil and duct tape expedition.
I can't relate to that as much. I guess when it comes to field work, every place that I've ever gone to collect fossils starts with a trip to the library. And that's, we are not just shooting into the dark here. We are building on the work that geologists, particularly stratigraphers, which are the kinds of geologists that look at the stacking of sedimentary rocks, how old the different sequences are, maybe something about the environment that they formed in. So every, field expedition first requires that kind of background. Then oftentimes it moves into aerial photos, and nowadays Google Earth. I mean, honestly, we can actually get the lay of the land and so forth. What we're looking for when we're out looking for vertebrate fossils are, of course, good rock exposures. Diana mentioned you can find them in urban or very, very remote places. I've tended to go to fairly remote places, although Pennsylvania road cuts, not around here, but all the way up in the north central part of the state, have been very productive for us. I'm generally looking for red rock. Once we get out to where this red Devonian rock is, then we've done our homework. It was formed in stream systems at a certain point in the Devonian period. I'm tending to look in the finer grained rock units, because those may be places where low energy deposits, in my cases, events that left sort of a muddy coating on the floodplain. So think of a high water event, and then that water starts to settle to go away, and it leaves a 4-inch pile of mud behind it. Well, that will stack up through time, and occasionally an animal will be dead in that mud. When we look at the sandstones, which are higher energy events that would have moved those sandstones, usually there are fish parts, but they've been through the wash. I mean, they're broken up. They're not as complete. We can often identify single elements and so forth, but what we like to find are more complete skeletons, or at least partially complete if possible. So we stand back, we read the rocks, we look at the different sorts of lithologies, so that's fine-grained versus coarse-grained. There's no substitute just for, it's not like you only walk, you know, a little this size of this room. We might walk 8 miles that way and find nothing, and then we'll walk 8 miles that way and find an outcrop where there is some fossil being eroded, and there might be more that we can find well preserved in the rock. So there's no substitute then for just getting on hands and knees, looking closely, walking to the next outcrop, to doing the work. It's not rocket science. I tell people that all the time. Anybody in this room, we could say, this is the kind of stuff, this is how you tell rock from bone, in our case. Let's go find some. And you'd all be great at it. Yeah.
I think just to echo what Diana and Ted have said is field geology is this interesting combination of low-tech duct tape, tin foil, you know, like scrambling to find a gas station, kind of work, but it also has this kind of ear training, art history, kind of like deeply sort of intellectual exercise of like trying to match things, trying to differentiate different kinds of beds and find distinctive structures. And then once you've seen it and you've locked in on it, you can recognize it everywhere. And I fell in love with geology as an undergraduate student and spent my first field project scrambling up and down these wet cliffs in Ireland looking for spores of the earliest land plants, just hauling rocks up and down in a backpack, completely oblivious to the danger of doing that. But you know, that's why you get 20-year-olds to do this kind of work. And so I've it's part adventure, it's part boredom, it's part incredibly exciting. You have these days where you find something, you turn around and you've read something in a paper, and then you turn around and you realize that on the other side of the canyon there's this incredible formation that the person was just standing here, locked in here, but didn't turn around. And I've had that experience before, which is really amazing, because you're like, oh, I've got something new. But lately, actually, one of the things that I've been doing that is kind of a slightly different version of field work is going into museum collections. And for plant material in particular, we have this sort of double-edged sword, which is there are particular times in Earth history, especially in the Paleozoic era, where plant material is preserved in these minerals that are highly rich in sulfide. And so they will, over time, decay. The sulfide will get oxidized, they'll turn into sulfuric acid, and the fossils themselves will degrade in a drawer. And we have 1.5 centuries of this material collected up and down the East Coast, but it's literally falling apart in museum collections. So I spend a lot of time now basically going to museum collections and digging out through these unmarked drawers. Like, I love a good junk drawer. I always tell a curator, like, show me where's the junk drawer that nobody's opened since the 1950s. Like, that's where I'll start. And pulling out fossils and trying to take something that is, probably not long for scientific knowledge and kind of bringing it into circulation. What can we do? Can we do some geochemistry with this? Can we do some morphometric analysis with this? Can we do the kind of physiological work with it? And that's its own kind of interesting prospecting. For me, it kind of rhymes a little bit with field work, where you pull open a drawer, it's just like turning a corner on a road. You never quite know what you're going to find next.
That's so true. I mean, I think the history of collection has outpaced the history of research performed on those collections. And so there is so much work that can be done just by opening cabinets as opposed to going out into the field. And then there's always new techniques and new technologies that can be applied to old specimens in order to say new things about old data. So thank you so much for this conversation. This has been a lot of fun. I think we want to make sure we can include audience members into the conversation. So maybe we'll now turn things over to you, and please raise your hand. We have a microphone which can come around in order to get your questions heard. Thank you, Desta. Sure, yeah, yeah. Yeah, so that's a good question. The question is, if you were to go back to the Devonian period, maybe different periods of time within that geological period, the question is, would you be able to breathe? Is there enough oxygen available?
This is actually an active area of research right now because we don't have a direct proxy for atmospheric oxygen. We have indirect proxies that give us a sense of the range. And so this is actually where I run into my human physiology limitations. I think we're probably looking at, what, 10% oxygen in the early Devonian, maybe slightly more. Does that seem like reasonable? I think that would be hard. I feel like that would be stressful, but let me turn this over to the vertebrate paleontologist. How do animals fare at 10% oxygen?
Never experimented on that.
Yes, they do. We have a fairly good record with independent lines of evidence that suggests that there's a dramatic fall in carbon dioxide near the close of the Devonian that's related to this mass extinction in the ocean, and there's the oxygen level starts to rise near the end of the Devonian, reaching a peak that's actually greater than it is today, we think, during the Carboniferous period, which follows the Devonian. So you probably wouldn't be able to breathe. It would be at the early Devonian, but by the latest Devonian, I think it might be like being at very high altitude.
Yeah, and it's important to note that photosynthesis predates the Devonian by a considerable amount of geological time. So this isn't the start of sort of oxygen contribution to the atmosphere as a result of photosynthesis, but certainly there is an uptick when plants start to cover the terrestrial spaces that haven't been covered. Yeah.
Yeah, so I have never heard of plant physiology before. That's so cool. So, yeah, how is that done? Do you do CT scans on the plant fossils and make a model or something? So, yeah, explain that to me.
Sure. Yeah, so plant physiology is just the sort of field where we assess the sort of physiological capabilities of plants. And one of the unique things that we do in my lab is we are basically able to quantify the hydraulic resistance of the vascular system, the drag. How easy is it for water to move through the plant from the soil to the site of photosynthesis. And the way we do this is we're lucky because the resistant parts of plants, the vascular system, it's made up of a biopolymer called lignin. And lignin is what makes wood resistant, but it also makes it preserve extraordinarily well in the fossil record. So we have these beautifully lignified vascular systems in the fossil record. And for my research, starting with my dissertation up until today, we build fluid dynamics models of flow through the tubes. And so we take a plant fossil and we do one of two things. We either take pictures of it and measure it and basically measure it to make it, is it the size and shape of a boba tea straw, or is it more like a coffee stirrer? Or, which is slightly more fun, we can take a fossil and we can drop it in acid, and all of the easier material, the more labile material, will basically dissolve away. But the lignified tissue, the vascular system, remains behind. And so we can take that out, we can pipette it, we can image it, and my students, my undergraduate students, make measurements of hundreds or even thousands of these so we can quantify the drag through the hydraulic system, which is a core constraint on how quickly the plants could grow, how fast their photosynthesis could occur.
Jason, you had mentioned some of the newer technologies in passing, and I wonder if the panel can talk a little bit about what the new technologies are. I'm curious in general, but also I wonder how you're able to detect color, for example, colors of vertebrates and maybe colors of plants.
Yeah, so that's a great question about new technologies being applied to fossils, and also whether color can ever be apparent in a fossil plant or animal.
I could start with some technology, although I'm kind of a Luddite. So I count on the younger generation to be managing the technologies. In vertebrate paleo, CT scanning has become very, very prominent. So a CT scan, of course, is a series of X-rays. Even if the fossil is still embedded in rock, or part of it is still embedded in rock, or if there is rock, what was silt or mud or whatever it might be within the body cavity and the skeleton, or even within the bones themselves. you can differentiate the bone from what is now the rock material. And it's very labor intensive, but each of these X-ray slices can then be sort of, you can cut out the rock and keep the bone, and then the computer will assemble all of the bone pieces together. You can do things like see the vascular canals that would have been filled with rock. You can just do so many things that we didn't think were possible. Not to mention that it also allows you to they call it animate, or basically visualize things, not only for education and exhibition, but also for the scientific literature, really to show off details that you really couldn't document just from some sort of photograph, or what we call a thin section, you know, a slice through a fossil bone or a tissue of some sort. So CT scanning, and to take CT scanning to the next step, which, or it's not CT scanning, but synchrotrons, which use very high energy particles to get down. One nice thing about CT and synchrotron work is it's non-destructive. We don't have to cut the bones up to do this. You can actually get down to the cellular level with the synchrotron, and there's only a few facilities that have that capacity, but it's being used now in paleontology. So this we don't have a lot of museums do have CT scanners now. They're expensive, and they're expensive to keep and to have staff to do. So we're not quite at the scale that can do that. Although the university over at Drexel, they got a grant and they have a CT scanner, which we've been throwing some fossils in. So it's a great opportunity.
And to your color question, so the feathers of dinosaurs, it's melanin, are the proteins, the shape, the alignment of particular proteins when the feathers are preserved are diagnostic of color, which is, I feel like it's kind of magic. I understand it, but it's also like amazing that these things are extinct. We can know some of the coloration. And certainly the imaging, I have not gotten to use it, but there are CT scans that can be taken out into the field, and you can, for me, I like the reefs too, and so there's some of these stromatoporoids that are digitate, and so scanning or imaging an outcrop and looking at the distribution, I want to get one of those if you have enough money for that.
Toys.
Great.
Earth and anaerobic systems that are going to predate these oxygen-driven systems, and they're what's the timetable and where are things when this Devonian period starts? What's the overlap? And is a lot of that still driving everything when the Devonian starts, or can you say anything about that?
Yeah, so you're asking about anaerobic systems on Earth before the rise of oxygen that we see in the late Devonian. Yeah, this is so as Jason said, photosynthesis is ancient. Photosynthesis goes back more than 2.5 billion years. But it's the terrestrialization of photosynthesis that really does seem to have a huge change on the Earth's elemental cycles. And if you think about that, if the photosynthesis has been occurring in the ocean, we essentially, we increase the habitat by an extra 30% by putting plants on land, and then they start to get complex. So we do see a lot of anaerobic environments through the Paleozoic. There's a lot of famous invertebrate deposits that I'm sure Diana could speak to. But we see that the Devonian is kind of this tipping point, where at the beginning of the Devonian and in the Ordovician and the Silurian that precede it, we get little scraps of things that look like early algal to land plant transitions, but they're not very abundant, and they appear to be restricted to a sort of narrow latitudinal range. Unusually, they all appear to be very close to the South Pole in the Ordovician. It's one of these enduring mysteries of, is that just where our record is best, or was there something going on in the Southern Hemisphere? But by the time you get to the Devonian, and especially as you transition to the Carboniferous period, we start to see things that look, that wouldn't be unfamiliar if you've been to the Fakahatchee Swamp in Florida or the Birdfoot Delta of Louisiana. You start to see things that look like those kinds of rainforests, but the composition is completely different. Instead of large flowering plant trees, it's the giant lycophyte scale trees. You have giant horsetail trees there as well. So the anaerobic environments never go away. There's always places on land and in Earth where For a variety of reasons, oxygen is low, but things really do seem to tip in the Devonian. And it's sort of, we go into a more oxygenated aerobic world.
We'll take one more question, then we'll have to finish up.
You were talking about big extinction events. I'd love to hear a little bit more about that. What went extinct? And do we have any idea why?
So this question is about the end Devonian extinction. What went extinct and what hypotheses exist as to why the extinction occurred?
Yes, and so this is part of the reason why I love the late Devonian mass extinctions. There's no singular, definitive, clear answer. It's still a little bit of a mystery. But I'll tell you what we do know. Trilobites might be some of the most famous critters that get hit. A huge number of them go extinct. Stromatoporoids, so many of my favorite brachiopods. The marine ecosystem, don't laugh, it's sad. But yeah, so a lot of these, especially tropical marine groups, are just decimated at this time. And I'll tell you what we do know about what's happening. There is dramatic climate change, but it's not an instantaneous change. There is drawdown of carbon dioxide due to the pesky plants, photosynthesizing, but the timing isn't quite right. So I think this change in climate, this overall decrease in temperature is causing just enough destabilization that even local perturbations probably were enough to cause extinction. And there is not a singular event. There are two big bad extinctions and at least a half dozen smaller over 25 million years. I don't know, what am I forgetting?
Since people then often do think about vertebrates and extinctions, I have to say that it's actually a good example of, I would say, limitations of the fossil record when I try to think about late Devonian extinctions. And we have, because as Diana said, when you talk about the 5 big extinctions, if you're not including the 6th that's going on these days, when you talk about the 5 big extinctions, the Devonian is there. And I think to myself, wow, we look at that time frame. What's wrong with us? Why don't we see change? And of course, we're looking at the freshwater ecosystems, and we're looking at the vertebrate animals there. But our record has very low resolution. And that's not always what people think about when they think about the fossil record. There are some organisms, particularly marine organisms, where you can actually see what happens every, I'm going to make up 100,000 years or something through time. You get another sample and another sample in an ecosystem that has changed or has not changed, and you can kind of come up with the scenarios for why that change was happening, what was coming, what was going, and so forth. With the vertebrate record, we have samples that are separated both geographically and by time, and it's very hard for me to find, we can talk about our Red Hills site, and it is approximately a moment in time, or at least geologically it's 10,000 or 50,000 years, which is a moment in time geologically. But how can I compare the animals that were alive there to another deposit up in Canada, which is, approximately the same age, but I'd have to say give or take 2 million years. So we can't ask questions with the vertebrates that require resolution on the kind of time scale to recognize these extinctions. And it's a frustration, but it's the reality. And the same is true for a lot of terrestrial fossil vertebrates. If you're going to study evolutionary patterns and extinctions, study brachiopods, don't study dinosaurs, for sure.
Yeah, I think for plants face the same problem as vertebrates. We have these beautiful deposits that are spectacular in their morphological diversity, but they're few and far between. And so if you're trying to sort of put a pin in the causality, is it the evolution of root systems that are deeper than X meter that then causes this systematic change that then tips the ocean over into this anoxic state, you really need to know time. And it's difficult for plants, but I think there is hope out there that, you know, maybe some graduate student is gonna walk around the other side of the canyon and discover one someday, and that'll help like shed some light on this problem.
Great. Well, Molly Gross is going to make some final comments, but I'll close this part of the program by just saying that this has been a real honor for me to moderate this fascinating conversation. So please join me in thanking our three speakers.
And thank you to you, Jason, for moderating it so beautifully. This was what we had hoped, right? To fill in a really rich picture of this fascinating period of time. And thank you all for your amazing teachers. So it was really wonderful. And you were a fantastic audience with your excellent questions. So thank you all.
Thanks, everybody. I'm Molly. I'm the public programs manager here, and we're just thrilled that this all came off so well and so beautifully. What I wanted to do, besides thank you for coming, is there's going to be another town square just next week. So next Thursday, October 3rd, it's going to be Planting for the Future. It's a conversation moderated by Mike and Scott from WHYY. You're going to hear from Chris Bolden Newsom. He's the farm manager at Sankofa Farms from Bartram's Garden, Owen Taylor, who's the owner of True Love Seeds, and Iris Brown from Villa Africana Colombo, and Chelsea Smith, who's the manager of the botany collection here. In this conversation, you're going to learn from the community members, farmers, seed keepers, and scientists that are working together to preserve the plant world right now, and that we all rely on, and that you can connect to our ancestral roots. So this town square is in conjunction with Heirloom Plants, Ancestral Seeds in Philadelphia. It's the exhibition on view now in our Spotlight Gallery through February 17th. So come back next Thursday, and we'll see you again. Thank you so much.